5.3 Mitosis. Mitosis is nuclear division in somatic cells, so that's going to be body cells. Mitosis occurs for growth and for repair. And remember, we are going to maintain the chromosome number. So one diploid cell is going to divide and produce two diploid daughter cells. So one cell becomes two, and we're not going to change the chromosome number. This is possible because if you remember back during the S stage of interphase, all of the DNA was replicated. So we're going to use these little diagrams to represent either a chromosome or a replicated chromosome. So on the left, we see a single chromosome, very highly condensed, highly coiled DNA. During S of interphase, that gets copied. And so we use this little X structure to represent a replicated chromosome. So now both of these copies are called sister chromatids. They're exact copies of that DNA. And these sister chromatids are held together at a point called the centromere. So an overview slide of what we're going to be looking at of mitosis. We start with a parent cell up at the top. You see the diploid number 2n equals 4. So this is two pair of chromosomes. We have a short pair and a long pair. Okay, those are replicated during S of interphase. And so in the middle diagram, we see these X structures. Both of the short chromosomes have been copied. Both of the long chromosomes have been copied. So now we have these sister chromatids that are held together at the centromere. And during mitosis, these are going to simply line up in the middle of the cell, and we are going to separate those sister chromatids. So we end up with two daughter cells, and if you look down at the bottom, you see two daughter cells. The diploid number is 4. Both of the cells at the bottom have one long pair of chromosomes and one short pair of chromosomes. Now, mitosis occurs in some stages. And so the stages of mitosis that we will look at are prophase, metaphase, anaphase, and telophase. This diagram shows what happens during prophase. Early in prophase, the chromosomes have already been duplicated, and so they condense into the very visible chromosomes. And also the nuclear envelope begins to break down. If we're going to divide the nucleus, we have to get rid of the nuclear envelope. If you look at the picture up at the top, this is an animal cell, and in the center you see the nucleus, and the darker stained material is the DNA. And initially you see it is relatively evenly dispersed throughout the nucleus. It's beginning to condense into some more visible strands. As we move further into prophase, the nucleolus disappears. The chromosomes become even more highly condensed, and they are going to start moving towards the center of the cell. Also, the centrioles have been replicated, and those centrioles begin moving to opposite poles. Late in prophase, this is over on the right-hand diagram, you see that the paired centrioles are on opposite poles from one another. The sister chromatids all are condensed. Those begin to move towards the center of the cell. And there are some fibers. These are called spindle fibers. They're microtubules. They're made of microtubules. And one of these spindle fibers is going to extend from the centrioles on the ends and attach to those sister chromatids. Remember from our overview slide, what we want to do is pull these sister chromatids apart. So for each pair of sister chromatids, one's going to get pulled to the left, one's going to get pulled to the right. And so we get these spindle fibers that radiate from the centrioles attaching to the sister chromatids at the centromere. Metaphase is the second stage. Metaphase is the easiest to identify. What you see in metaphase is those sister chromatids are all lined up in a straight line. This is called the metaphase plate. The spindle fibers now are all in place. They radiate from the centrioles attached to the centromere. If you look at the picture up at the top, again, this same animal cell, you see that straight line across the middle. That is the metaphase plate. During anaphase, the spindle fibers begin to shorten, and this is what actually pulls those sister chromatids apart. So as we mentioned, we're going to separate each pair of sister chromatids. So this occurs in anaphase. And then the last stage is telophase. In telophase, we have completed separating the sister chromatids. The nuclear envelope is going to reform. The nucleolus is going to reform. And also during telophase, we begin cytokinesis. Cytokinesis is what's going to divide the cytoplasm and the organelles of the cell. And so this begins with the appearance of a cleavage furrow. It's kind of like pulling a drawstring around this cell and pinching it in half. This is a diagram that shows mitosis in plant cells. Plants go through the same stages. Prophase, you see the condensing chromosomes, the nuclear envelope has disappeared, the nucleolus has disappeared. In metaphase, we're going to line the sister chromatids up on the center line there in the cell. That line's not as striking as what we saw in the animal cell. It's not that straight line, but still they're going to be lined up in the center. Anaphase, we are going to pull the sister chromatids apart, and then telophase, we reform the nuclei. The final step in plant cells is also cytokinesis. Remember, a plant has a cell wall, and so we have to additionally reform the cell wall. So in the diagram on the far right, you can see indicated the cell plate, and this is where that cell wall is beginning to reform. Here are some pictures that just illustrate cytokinesis a little bit. This is done with a scanning electron micrograph. And so up at the top, this would be an animal cell. You can see the formation of the cleavage furrow. This cleavage furrow gets deeper. Eventually, these two daughter cells pinch, come apart. And as we saw in that previous diagram, we have to reform the cell plate. This is a transmission electron micrograph of the cell plate or cell wall reforming in plant cells. Before we move on to meiosis, we'll take just a moment and look quickly at cell division in prokaryotes. Do prokaryotes go through mitosis? Remember, mitosis specifically is nuclear division, and prokaryotes lack a nucleus, and so they go through cell division, which is called binary fission. Prokaryotes have a single circular chromosome. That chromosome is replicated. The cell then elongates and pinches into, in a process very similar to cytokinesis. So here are some simple diagrams that show this binary fission in a prokaryote. That single circular chromosome gets copied, the cell elongates, pinches in half. We have two new prokaryotes. Animal cells, mammalian cells, for example, can go through the cell cycle in roughly 24 hours. So they complete interphase and then mitosis. Mitosis takes several hours. A prokaryote can divide in 20 or 30 minutes. So much simpler process, much more rapid cell division.